Hello, my name is Michaela Rodriguez, and I am a senior at UT Permian Basin pursuing a Bachelor of Science in Biochemistry. I am currently studying in the research lab of Dr. Xu, Hello, my name is Michaela Rodriguez, and today I will be presenting my project titled Determining How Psap Induces Mitochondrial Dysfunction. Again, my name is Michaela Rodriguez, and I am a senior studying biochemistry at UT Permian Basin in Odessa. I am currently studying in the Neurodegenerative Research Laboratory of Dr. Xu using a live animal mouse model. Dr. Xu's lab focuses on the molecular mechanisms involved in neurodegenerative disorders such as Alzheimer's and ALS. Psap protein, which stands for Presnicillin-associated protein, has been identified as being a pro-apoptotic protein, that induces programmed cell death or apoptosis. This project aims to determine the way in which the protein Psap induces mitochondrial dysfunction leading to programmed cell death. Hello, my name is Michaela Rodriguez, and today I will be presenting my project titled Determining How Psap Protein Induces Mitochondrial Dysfunction. My name is Michaela Rodriguez, and I am a senior at UT Permian Basin pursuing a Bachelor of Science in Biochemistry. I am currently studying in the research lab of Dr. Xu, focusing on Alzheimer's disease and ALS, or more commonly known as Lou Gehrig's disease. Dr. Xu's lab focuses on understanding the cellular and molecular biomechanisms that lead to neurodegenerative diseases like ALS and Alzheimer's using a live animal model, or Dr. Xu's lab focuses on the cellular and molecular biomechanisms that are involved in neurodegenerative diseases such as Alzheimer's and ALS. His lab uses a mouse model. Dr. Xu's lab focuses on understanding the cellular and molecular biomechanisms that are involved in neurodegenerative diseases using a mouse model. Psap protein has been observed to implicate the pathogenesis of multiple disorders such as AIDS, Alzheimer's, ALS, and more. My name is Michaela Rodriguez, and I'm a senior at UT Permian Basin pursuing a BS in biochemistry. I am currently studying in the research lab of Dr. Xu, focusing on Alzheimer's disease and amyotrophic lateral sclerosis, or ALS. Dr. Xu's lab focuses on understanding the cellular molecular biochemical Hello, my name is Michaela Rodriguez, and today I will be presenting my project titled Determining How Psap Induces Mitochondrial Dysfunction. Again, my name is Michaela Rodriguez, and I am a senior at UT Permian Basin pursuing a Bachelor of Science in Biochemistry. I am currently studying in the Neurodegenerative Research Laboratory of Dr. Xu, focusing on Alzheimer's disease and ALS using a live mouse model. Dr. Xu's lab focuses on understanding the cellular and molecular biochemical mechanisms involved in neurodegenerative disorders and diseases. Presnicillin-associated protein, or Psap, has been identified as being a pro-apoptotic mitochondrial protein. PSAP has been observed to induce programmed cell death or apoptosis, implicating the pathogenesis of multiple disorders like ALS and Alzheimer's. This project aims to determine the way in which the protein PSAP induces mitochondrial dysfunction, leading to programmed cell death or apoptosis. This was accomplished using a cell line, HEC293, which stands for human embryo kidney cells, which have been stably induced by a plasmid that expresses PSAP protein. Upon the addition of ponesterone A or PON-A, a gene or PON-A, an inducer, or PON-A, a gene expression inducer, upon addition of PON-A to the HEC293 cells, the expression of PSAP was induced, leading to the expression of PSAP was induced, ultimately leading to apoptosis. Subsequent to the addition of PON-A to the HEC293 cells, apoptotic expression was measured via the detection of specific proteins. Subsequently, the apoptotic expression of the HEC293 cells induced with PON-A were measured via the detection of specific proteins using the Western blot methodology. In addition to the cellular and molecular components of this project, animal behavior was studied using a mouse model of ALS transgenic mice, which have been bred to express SOD1693A, the gene for SOD1. In addition to the cellular and molecular components of ALS and Alzheimer's, animal behavior and welfare was assessed. In addition to the cellular and molecular components of this project, animal behavior was studied using a mouse model of ALS transgenic mice. Specifically, the grip strength of mice was assessed using a grip strength meter in grams. The motor ability of mice was assessed using a rotarod apparatus, and body weight was tracked 3 times, and body weight was also assessed 3 times a week. Ignore. In addition to the cellular and molecular components of this project, animal behavior and welfare was also assessed throughout the 10-week duration of the this project. This component assessed the grip strength of mice, motor ability, body weight, and overall welfare and well-being of the animals on a regular basis. It is with great hope and enthusiasm that my sole mission in pursuing research science is to improve not only the lives of patients and individual humans impacted by these disorders, but the caretakers, families, and loved ones of these individuals as well. Materials and methodology. The HEC293 cells that were used in this project were previously purchased from a laboratory distributor and stored in liquid nitrogen for a year. The culture media that I used to maintain my cell line was DMEM, was DMEM, FBS, and a mixture of strep and penicillin. Ugh. No, I don't like it. The HEC293 cells were previously stored in liquid nitrogen, and then the culture media was also previously purchased. The culture media that I used to maintain my cell line was DMEM, FBS, and strep penicillin. The gene expression inducer ponesterone A was purchased from was purchased from in vitro gen and was distributed by Enzo Life Sciences. The gel electrophoresis power supply was graciously funded by the LSAMP program and was purchased through Biorad, specifically for use in my research project. Additionally, a molecular weight marker from Biorad was also purchased. Additionally, a kaleidoscope molecular marker was purchased as well. The rotorod apparatus and the rotorod apparatus and the grip strength meter were previously purchased by my primary investigator, and the HEC293 cells that I used for this project were previously stored in liquid nitrogen. Over a year ago. The culture media was also previously purchased by my PI, and I used DMEM, which stands for Dulbecco's Modified Eagle Medium, FBS, which stands for Fetal Bovine Serum, and streptomycin penicillin. The inducer, ponesterone A, or PON-A, was purchased from Thermo Scientific, but was distributed by Enzo Life Sciences. Additionally, the power supply used for my Western blots were purchased from Biorad and funded for by the gracious LSAMP SRA program. This was also, with this stipend, I was also able to purchase a kaleidoscope molecular marker that I used for my Western blots. The first component of my research project that I will be discussing will be the cell culture of HEC293 cells. HEC293 cells are a specialized cell that have been stably transfected with a plasmid that have been stably transfected with the plasmid that will express PSAP. These specialized cells were stored in liquid nitrogen for over a year. These HEC293 cells were previously stored in liquid nitrogen for over a year upon cell culture and upon being the first component of my project that I will be discussing is the cell culture of HEC293 cells. First, I needed to remove the HEC293 cells from liquid nitrogen and thaw them out very carefully as to prevent the cells from drastically changing, were carefully thought out as to prevent the cells from becoming shocked from a drastic change in temperature. The thawed cells were then placed in culture medium, prepared. The thawed cells were then placed in DMEM, FBS, and streptop. The thawed cells were then placed in the DMEM, FBS, strep penicillin mixture, strep penicillin culture media, and given time to grow. During this time, the cells were regularly observed for any visible or notable changes, as well as throughout the duration of this growth period, the cells were regularly observed under the microscope, and culture media was changed every 48 hours as to ensure adequate health of our cells. Once the cells were healthy enough and reached 80% confluency, we were ready to, and we're at 80% confluency. The cells were further split for use in. During this period, cells were regularly observed, and culture media was changed every 48 hours. Once confluent, the cells were then split into additional dishes. Some were going to be some to be used for the ponesterone A induction, and some for a stock supply. Next, the next step was to induce the expression of PSAP using ponesterone A. The next step was to induce apoptosis in HEC293 cells using ponesterone A. The HEC293 cells were seeded onto 60-millimeter dishes and given 24 hours to adhere upon the addition of ponesterone A. 45,000 cells were placed per dish following a cell count report. Once seeded, the HEC293 cells were subject to the addition of 20 micromolars of the PON-A inducer at a 1 to 1,000 dilution with DMEM. Subsequently, cells were harvested using a cell lysis buffer at 2. Subsequently, the samples were collected at 0, 2, 4, 6, 8, 10, 12, and 24 hours. This was accomplished using a gentle cell lysis buffer and stored using a gentle cell lysis buffer and then stored at minus 80 degrees Celsius until ready for Western blotting. Once all samples had been harvested and collected, I could begin my Western blotting. Figure one displays a general overview of the Western blotting process. Using the harvested cell lysates, I first sonicated my cell lysis and then quantified the concentration of my proteins using a BCA assay. Using the BCA results, samples were prepared according to dilutions. Using the BCA results, I calculated appropriate dilutions of proteins. Following the BCA assay, I calculated the appropriate dilutions for my samples, and then denatured the protein samples by boiling them in water for 10 minutes. While my samples boiled, while my samples cooled down, I began the process of setting up my gel electrophoresis apparatus and setup. The first step is to create your polyacrylamide gel pages, and each polyacrylamide gel page, or each gel membrane. The first step was to create my polyacrylamide gel membranes. Each of these membranes is specific to the protein of interest. the first step of setting up my apparatus was to create my gel membranes. Each gel membrane has a specific percentage of polyacrylamide in it, dependent on the protein of interest and its molecular weight. This process takes roughly 1 hour. Takes roughly 1 hour as to ensure the polymerization and solidification of our gel membranes. Once the gel membranes were solidified, they were placed into the once adequately solidified, the gel membranes were placed into the chamber and were placed into the chamber with running buffer as to ensure that the gel membranes do not dry out. Once the chamber was appropriately set up, the samples were loaded. Prior to loading protein samples, a molecular marker was loaded onto the first well. Then the samples were loaded according to time and ran at 70 volts for 4 hours. After the gel electrophoresis, I transferred my proteins onto a PVDF membrane and then blocked them in a milk blocking buffer for 1 hour. Following the blocking period, I began protein detection using a primary and a secondary antibody. Following my antibody staining, I subject my PVDF membranes to chemiluminescence and took images of them that I used for this project. Overall, Western blotting is a 2-day procedure and takes about 10 to 14 hours. This experiment is very, very time consuming and requires a lot of patience this experiment is very, very time consuming and requires patience and care. It is very easy to mess up one of many steps. And so I often had to redo my trials. Next, I will discuss my experience handling the mice and assessing their overall motor ability and welfare. Typically, I worked with about 8 cages of mice at a time. Each cage of mice had roughly 3 to 5 mice. 3 times a week, I weighed the mice with an analytical scale, averaging out the total weights of all the mice per cage. On the days that I did my weigh-ins, I also did an animal welfare check, which on the days that I weighed my mice, I also did an animal welfare check, which focused on conditions related to the hydration, appearance, activity, and possible clinical signs of illness in the mice. Once a week, I subject the mice to a grip strength meter in which I measured the grip strength of the forelimbs and then the forelimbs and hind limbs in grams. Again, an average value was taken for each cage of mice. 1 hour after taking my grip strength measurements, I put the mice on a rotarod apparatus. 1 hour after taking my grip strength measurements, I allowed the mice an acclimation period of 1 hour. Following the 1 hour acclimation period, I placed the mice on the rotarod apparatus, which assessed how fast and how long the mice were able to run on the track. This was done only once a week, with 5 to 7 days of rest in between each trial. The preliminary results of my Western blots reiterate the hypothesis that upon the addition of PON-A, HEC-293 cells will induce apoptosis via the overexpression of PSAP protein. This was confirmed using the Western blot methodology to detect proteins. This was confirmed using the Western blot methodology to detect proteins PSAP, PARP, and caspase-3, all hallmark to the apoptotic pathways seen in Alzheimer's and ALS. Unfortunately, not enough data was collected to make any inferences regarding the behavior of the mice. Not enough data was collected to make any inferences regarding the behavior of the mice. I can attribute this. Not enough data was collected to make any inferences regarding the behavior of the mice. One consideration to take into account is that working with mice is a lot more difficult than it sounds, and it is very time consuming and requires multiple days of training. It was a lot more time consuming than It was a lot more time consuming than I initially thought, and I attribute my lack of and I attribute the time component as being most influential to my inability to draw any conclusions based on the mice. The preliminary results of this project reiterate the hypothesis that the addition of PON-A to specialized HEC-293 cells induces apoptosis via the overexpression of PSAP. This was confirmed using the Western blot methodology to detect proteins PARP, PSAP, and caspase-3, which are all hallmark to the apoptotic pathways seen in ALS and Alzheimer's disease. Not enough data was collected to make inferences regarding the behavior of mice. One consideration to take into account is the time that goes behind handling mice, or handling animals in general. It is a very time-consuming project and requires multiple trainings, not only with the handler, but the mice themselves. In conclusion, neurodegenerative disorders are known to have complex and intricate mechanisms involving a multitude of interrelated cells and molecules. These mechanisms have continued to evolve with time. Therefore, it is imperative to continuously study and understand the mechanisms in which cellular and molecular dysfunction occurs. Supporting and developing presently existing data in science is equally, if not more, significant than the discovery of novel and unfamiliar findings in research. Lastly, I would like to go into my acknowledgements. This project received funding from the esteemed and prestigious Psap Summer Research Academy, serving undergraduates aspiring to attain careers in the research scientists. I would like to give special thanks to Dr. Milka Montes, the Psap director at UTPB, Dr. Xu, my research professor and mentor, and the Biomedical Research Center at UT Permian Basin for the continual support and guidance throughout my undergraduate education. Thank you.